So I'm Brian Browning from the University of Washington. I work primarily in methods development for haplotype inference and also methods that rely on haplotypes. So this can be genotype imputation, identity by descent segment detection, applications of IBD segment detection, and also most recently local ancestry inference. And I don't particularly work with compression methods, but I've been forced to think about them and deal with them a little bit because haplotype-based methods are most powerful with large data sets. We preferentially seek out very large data sets, and then you're confronted with the idea of how do you deal with this avalanche of data. So quick review of haplotype inference, or haplotypes, what they are. Usually the data we get is genotypes, so the line represents a chromosome. We have 4 sites here. We have a genotype at each site consisting of 2 alleles that are the alleles inherited from each of your parents. Haplotype inference tries to infer the sequence of alleles that are inherited from each parent. We don't normally get that directly from the genotype data, but using statistical methods, we can infer the sequence with high accuracy, if you have a large sample size, what the sequence of alleles are that you inherit from each parent. So here at each site, you can see you have the same alleles as before, but now we can see the A, C, T, and G we infer was inherited from one parent and the G, A, T, T sequence from the other. This is very suitable for compression. So if you phase your data, you should be able to compress it much better than if it's unphased because of the way genotype data is passed down. Chunks of DNA are passed from parent to offspring, and so people share large sections of DNA, so you have redundancy in the data in a large sample size, and you can use this redundancy to get very good compression, as you'll see. You might, just to start out, if you have 400,000 sequence samples compared to uncompressed text data, you might want to guess, and how much can we compress it down? And you'll get an answer at the end. Data sets are increasing because the sequencing costs are decreasing. A few years ago, it was a big event when we reached the $1,000 genome. Now we're approaching the $100 genome. At those costs, you can sequence very large cohorts. The All of Us project is looking to sequence 1 million individuals. The next release of data will have over 400,000 sequenced individuals. And I really don't see any reason why they're going to stop at 1 million. Why not go larger? Our Future Health in the UK is collecting 5 million samples. 5 million genomes, my back of the envelope calculation is if you store it in uncompressed text files in a format called VCF, which I'll explain on the next slide, you're looking at 15 petabytes, right? So it's mega, giga, tera, peta. So it's probably 6 orders of magnitude greater than any data set many of you have dealt with. But as you go on in your career, you may encounter, if you're dealing with large sample sizes, data sets of this size, at least in uncompressed form. You can use gzip to compress it, and that does quite well. It gets down to 80 terabytes, but that's still inconvenient, to say the least, to deal with. You spend a lot of time copying files, spend a lot of time waiting for bits to shift around. So the standard format that's used for analyses is variant call format. It essentially represents the data in a matrix format. It starts out with some header lines that begin with 2 hashes shown at the top. They're not very wide. They're typically less than 100 characters or so for each line, so they don't take up much space. Where the space is consumed is starting with the line that begins hash chrome. So that line contains labels for each column that goes below it. So the first 8 fields of this line describe information about a particular site in the genome. So each line is a site. So first we have the chromosome, the position of the site we're talking about. If you have an identifier like an RSNP number, you can throw it in the 3rd site, but typically it's a dot which represents missing data. The reference allele at that position, that's the alternate alleles, and you can have multiple alternate alleles, a measure of quality, a filter column that has pass if it passed all the sequence filters, or if it failed, it'll give a code that lets you know what filter or filters were failed. An info column, which is very useful for holding QC metrics. Here I've just put one in, which is the average allele depth, but it can have other QC metrics. And this column is very important if you want to filter on markers by minor allele frequency, or, well, actually by QC in general, QC measures in general. After these first 8 columns, you have a format column that tells you what kind of data you have for each sample. Here we just have the genotype data, and that's what I'm interested in. You can store other data per sample. If you do, it's not going to compress as well. Genotype data is highly compressible in general, or at least phase genotype data is. If you store other information like the number of alleles or number of reads that support each allele, you can store that here, but it doesn't compress as well. So in practice, I think you'll find it most useful to summarize any QC measures any measures that are taken on a per sample basis, get some mean, and summarize it in the info column so you can still use it for filtering, and you're only storing genotypes in the sample columns. So after column 9, you have sample identifiers in the top Chrome row. I just put ID1, ID2. You would put your data sample identifiers there. And you have genotypes for each sample. The 0 represents the reference allele. The pipe indicates the data is phased. And you can see you have 2 alleles for each individual. These files tend to be very wide. So if you have 500,000 samples, which is where we are now in easily readily available biobank scale data sets, you're talking 500,000 columns. All right, that's a very wide file. For a large sample of 500,000 individuals on a large chromosome, you probably have 100 million or so variant sites. So you've got 500,000 columns, 100 million rows. It's in a nice, easy to understand tabular format. You know, rows are markers, columns or haplotype or samples, but it's a big file. So how do you handle these large files today? Well, typically you compress them with bgzip, which is just a form of gzip compression that stores a little bit extra information to facilitate indexing. So with indexing, if you want to retrieve some data 3/4 of the way down the chromosome, it allows you to jump very close to where you want to retrieve the data and just start decompressing there, rather than starting at the beginning of the file and reading and decompressing everything to get down to that 3/4 point. So indexing is very important for dealing with large files. And the other common technique that's used by the UK Biobank is to shard. You shard the data into small genomic intervals. So the UK Biobank, I haven't looked at their most recent release yet, but I will soon. But the first 2 releases of data, they sharded into 60,000 files. Right? Each file represented about 50 KB of the genome. Now, dealing with 60,000 files is not particularly easy, but it's preferable in many cases to dealing with huge files that represent a whole chromosome of data. If you're analyzing on a per marker basis, you can just farm each shard of the data to a separate processor and analyze in parallel. So it facilitates that very nicely, and instead of taking the time to transfer some humongous file that represents a whole chromosome, you're transferring much smaller files. Very convenient for single marker analysis. If you're doing haplotype analysis, which is what I work with, it's slightly inconvenient because often with haplotype analysis, you do want to analyze whole chromosomes. And for those, then you have to merge the shards. So these are the current techniques that I see out there. So I want to survey some types of compression methods, and I'm going to evaluate them in two dimensions. First, of course, the amount of compression. How well do they compress the data? This affects how much it costs to store the data. It affects how much it costs to copy the data, which is quite common because if you're working in the cloud, you invariably have to copy data from cloud storage onto the virtual machine that you create to analyze the data. So copying, you can spend a surprising amount of time just copying data. Reading data in, reading the I/O time to read data in once it's copied to your virtual machine. Amount of compression affects all those things. And then the second thing we want to look at for these compression methods is what analysis can you perform directly on the compressed data? If you can analyze directly on the compressed data, it means you save the cost of decompression. You also save memory costs, because in memory, you can just store the compressed data. And because for some analysis in particular, not for all analysis, you can analyze, you're just iterating over the compressed data, your computations can go much faster because you're dealing with much less data, orders and orders of magnitude less data. So I'll look at 4 types of analysis or operations you might want to make on compressed data, and we'll look and see, is it possible using this type of compression method to perform this analysis, and thereby save time there. So the first, there's other analysis in this, but it's summary statistic type analysis, the most common of which is counting alleles. Right, counting alleles would allow you to do simple association tests. For example, it can be used for filtering data if you want to filter out low-frequency markers or retain only the low-frequency markers. Filtering samples. You have a big biobank. Maybe you want to look at a particular subpopulation. You don't want to analyze the whole biobank. You'd like to filter the samples. Can you do that on the compressed data without having to blow it up, inflate it, subset it, and then recompress it? Filtering markers. You might want to get rid of the low-frequency markers. Require very high QC metrics. Only look at SNVs. Only look at markers in a particular region of the genome. Can you filter markers just in compressed data without having to uncompress the data first and then compress it back later? And finally, can you reach in and grab an allele without decompressing the data? Or is it squashed in together and compressed so much that there's just no way you could figure out what that allele is without actually blowing it up and inflating it again? So those are the 4 types of analyses we'll consider. When you think of compression, often there's a concern about the cost of storage. Like, I've encountered this by people who work in computation. They're concerned about the cost of storage. Well, actually, the cost of storage isn't that bad. Even with gzip, we'll show that there's even better compression methods than gzip in terms of how much you can compress the data. But even with gzip, my back-of-the-envelope calculation is for 5 million genomes. So we're at about the 500,000 genome point now. In a few years, we'll be in 5 million genomes. Even in a few years, it's still only around $10,000 to store these data. And you typically only have to store 1 copy. A bigger cost tends to be the analysis. Let's say you want to filter that data. Well, decompressing the data for filtering it and compressing those 5 million genomes in gzip format takes about 6.5, a bit more than 6.5 years of compute time by my estimate. Now, of course, you're not waiting for 6.5 years. Start at the end of your PhD and get your answer back at the end. You do it in parallel. And so the wall clock time may be measured in minutes, but the compute time is significant. And depending on whether you use spot instances or on-demand instances, the cost of just this one operation for one analysis can be between $1,000 and $6,000 or $7,000. So imagine, you have 1 copy of the phase data and all these groups doing analysis of this phase data. The analysis cost will dwarf the storage costs. So we'll look at 3 types of compression methods. These are not the only 3 types. April Way gave a talk in this CGSI session, or the CGSI week, using a compression method that's inspired by the ancestral recombination graph. So there's others out there. I'm just going to look at 3 that I'm most familiar with and that have been around for a while. General compression methods like gzip, IBS, or identity by state segment methods that make use of the fact that haplotypes share sequences of alleles to compress the data. And examples would be M3VCF and BREF3. These methods were originally developed for use with imputation methods, where you use them to compress a reference panel. And then PBWT methods, where PBWT is the positional Burrows-Wheeler transform. We'll talk about, if you've heard about that and wondered what it was, we'll go through what it does to the data and why it can be used for compression. So gzip format, it's a wonderfully successful format because it's not that hard to work with. It's integrated into all our tools. If you want to change from reading or writing a text file to a gzip file, typically it's changing 1 line of code. Very easy. cat, if you want to look at what's in a gzip file, you just, instead of using cat at the Unix prompt, you use zcat. It's very easy to work with. And it consists of what the specification calls members. The specifications are listed here. The RFCs 1951 and 1952. Starts with 10 bytes, at least 10 bytes of header data, then the compressed data, and then 8 trailing bytes. So it consists of these blocks. And this gzip is a prototype black box. You don't have to know how it works, but you just use it. It's wonderful. We'll look at a little bit how it works, just so you understand why it does a fairly good job compressing sequence data. Its advantages are that it can be applied to any byte sequence. It's widely installed. It gives good compression. Disadvantages if with really large data sets, sometimes you need a bit more. Good isn't good enough. You really need state-of-the-art compression. And decompression is required for nearly all operations. That's a nuisance with gzip. One exception is concatenation of files, and this is an important trick to have in mind. Because gzip consists of a sequence of blocks of compressed data, if you take 2 gzip files and just catenate them, don't decompress them, just in their compressed form, just catenate them with the Unix cat command, you get a valid gzip file. That's very valuable if you're working with sharded data and you need to merge the sharded data. So it's a useful trick. But that's about all you can do with gzip data without decompressing it. So here's an example of how gzip can compress sequence data. So here we have some VCF text from a VCX file. The 0, vertical pipe 0, represents a homozygous reference genotype. And because most markers, when you do sequencing at very low frequency, you get long strings of these homozygous reference genotypes. And the underscore just represents the tab, which is the delimiter used in VCF files. So this string of 4 homozygous reference genotypes compresses to something like what I've shown here on the second bullet point. You get 0, vertical pipe 0, tab, and then a code. And that code consists of 2 numbers, and it tells you copy character from 4 places back and copy it 11 times. So I've given an example of how that works. So first you copy the 0, vertical pipe 0 pipe, and then you get this code that says start copying stuff that came back before. So first you go back 4 characters, you get the initial character 0, you copy that. Now that you've copied it, your leading edge has advanced 1, so when you go back 4 characters, it's also shifted 1 to the right. You copy the pipe. Then you copy the next 0, and you do that 11 times, and voila, you get back what you started with. You've decompressed the data. Now do a thought experiment. You have 500,000 genotypes, right? Data set with 500,000 genotypes. You're at a low frequency marker, which is probably most of the markers in your data. This low frequency marker has a stretch of 250,000 homozygous reference genotypes. Can you see how you'd collapse that using gzip? What changes, considering our compressed section, where we had 4 homozygous reference genotypes, if instead we had 250,000, what would change about our compression? Just that last number. Instead of 11, we'd have some number around close to 1 million, right? Because 250,000 homozygous reference genotypes, each genotype has 4 characters to express it in text, so you're talking about 1 million characters. It compresses wonderfully well, right? It's just that last value changes and collapses down. Or, the last value changes from 11 to something just under 1 million. So you get very good compression of repeated homozygous reference genotypes, and as a result, we're going to see gzip does very well. It compresses, we'll show you some data that shows it compresses by a factor of 300. 300 bytes on average get compressed down to 1 byte. Very good, but sometimes not good enough. Next, M3 VCF compression. The idea here, these methods are IBS-based. You just partition a chromosome into non-overlapping windows, and in each window you store the unique sequences and store a pointer from a haplotype to the sequence it carries. So a picture will make this clearer. So in this toy example, haplotypes are rows on the left. There's 6 haplotypes labeled H1 through H6, 47 markers on top labeling the columns. The green rows indicate these haplotypes 1, 5, and 6 all carry the same allele sequences. The yellow rows carry the same allele sequence, and then there's a third allele sequence carried only by haplotype 3. So instead of storing all the allele sequences in this window, we just store the unique ones. So we just store 3, and you can see the compression. Instead of a matrix of 6 times 6 rows and 47 columns on the left, we store a matrix of 3 rows and 47 columns on the right. So there's compression. The cost is you have to store the pointers. So the pointers, you can see they're a box with a color that just says, for this haplotype, it points to the allele sequence it carries. So it's lossless compression. You can look up. You haven't lost any information, but it stores it in a much smaller format. I'll just mention BREF3 compression. It's the same as M3VCF, but it peels off the low-frequency markers, and it just stores the haplotype indices that carry the minor allele. And this is also a lossless form of compression. So if just 2 haplotypes carry a minor allele at a marker, 52 and 998, you just store those numbers, and if you want to, given a haplotype like 75, and you want to see whether 75 carries the minor allele, you just look at your list. If your 75 is in the list, in this case it's not, it must store the major allele. But if 75 was in our list, you know it stores the minor allele, so it's lossless. And then once you compress the low frequency markers that way, the high frequencies are handled the same way as M3VCF. So the advantage of these methods, and I don't think people have looked at them very much this way, but they have a lot of promise because you can analyze data without decompression. You can store the memory, you read it in compressed format, you store it in compressed format in memory. You don't have to decompress it at all. And then you can access it as if it were a matrix, as if you had decompressed it into a large 2-dimensional matrix. So the pseudocode, an example down here, is get a marker and a haplotype. Marker would be like you're accessing a row of a matrix of alleles. Haplotype would be the column of the matrix of alleles. It looks like you're accessing a 2-dimensional matrix, but looks can be deceiving. This is just an interface. You don't really know what's happening behind that interface. And behind that interface, instead of 2 array accesses, which is what you'd need to access a 2-dimensional matrix, there's 3 accesses going on if it's in M3VCF format. 1 array access goes from the marker to find the window it's in. 2nd array access goes from the haplotype to find the unique haplotype sequence in the window. 3rd array access pulls out the allele on that unique haplotype index. And although there's an extra array access in practice, it should perform very well because the data is more compressed. The data you need is more likely to be in cache, and so you have fewer cache misses. And anytime it's not in cache and you have to go to main memory, that's actually a very expensive operation. Amount of compression, we'll see with real data at the end, is better than gzip, but worse than PBWT with run length encoding, which we'll look at next. PBWT is positional Burrows-Wheeler transform. It sorts alleles at a marker by their reverse haplotype prefix. So what it does is you look at all the alleles that come before on the haplotype, and you sort them, dictionary sort, in reading from right to left. We'll give a picture in just a minute. And then after that sorting, where you've reordered the alleles at the marker you're interested in because of the sorting, you apply run length encoding. And the first time you see this, you might wonder, is this reversible? Is this really a lossless compression? The answer, it is, but it's very natural to wonder if it's reversible, but it is. The paper, if you work at all with haplotypes, I highly recommend reading the Durbin bioinformatics paper. It's a significant paper in the field. So what is reverse prefix sorting? Here, we're interested in compressing the data at a marker that's not shown. I'm just showing the prefix to the marker, all the haplotypes that come before. So haplotypes here are rows. This is an illustration from Durbin's paper. And we don't have the time for you to stare at it long enough, but these are sorted in reverse prefix order. So they're sorted right to left. 0 comes before 1. It's easiest to see if you look at that last column. If you look at the last column, you can see the 0s in that last column occur before the 1s. That's just like a dictionary would do it. But again, we're reading right to left. So trust me, it's sorted right to left, or at least I've checked a few, and Durbin claims it's sorted, and I haven't exhaustively searched it, but it should be sorted. So once they're sorted, that means haplotypes that are next to each other in the sorted order are likely to contain long sequences of shared alleles. And if 2 haplotypes have a long sequence of shared alleles, they're more likely to share an allele with the next haplotype. So now that you've sorted the prefixes, I've added a character that shows the alleles at the next marker. And so if you notice in this toy example from Durbin's paper, the first 8 rows all have a 1. Now that they're sorted, it turns out that at the marker we're interested in, we got a big row sequence of 1s together. And then later on, we have a sequence of 5 zeros. So it becomes amenable to run length encoding. Instead of storing 8 copies of 1, you store a 1 once and then a number that says how many times you copied it or how many times in a row it appears. So you'd store a code that says, the allele is the allele that's stored 1 and how many times it appears. And down below, there'd be, for the 5 copies of 0, it'd be 0 is the allele being repeated, and it's repeated for a total of 5 copies. So that's where the compression, the PBWT just makes the data more compressible for something as simple as run length encoding. Advantages, excellent compression. That's, if you look at the papers coming out, proposing new methods, they often use the PBWT because it gives very good compression. Disadvantages, most analyses require decompression. There are some that you can do, but some important ones that you cannot. You can't filter by marker. If you filter by marker and exclude a marker from the compressed data, you've just lost the ability to decompress anything after it. All right, not a good property. Retrieve a haplotype allele, again, you're going to have to decompress. If they use some kind of block approach, you're going to have to decompress a whole block to get an allele. Again, you have to go to a lot of work. But trade-off is you get very good compression. Now I want to talk about a method. This is unpublished, but it's getting close to getting out there to publication, called BREF4. It's the follow-up method to BREF3 that tries to give you the best of both worlds. It tries to give you ability to do analysis directly on compressed data so that you never have to decompress the data and approach or come very close to PBWT compression. Can you have your cake and eat it too? So it starts out just using BREF3 compression. The low frequency markers, you just store the haplotypes that carry their minor allele. Then high frequency markers you compress by breaking into windows and storing the unique sequences on each window. So this is just like M3VCF or BREF3. But rather than stop here like those other methods do, you then repeat, sort of recurse. You have a window of unique haplotypes, and now you say, well, let's pretend this window is a whole chromosome. Let's pretend these unique haplotypes in the window are the haplotypes I need to compress, and you just repeat the compression. You break it up into windows, and the idea is once you break it up into windows, that haplotypes that on the left are unique in the window, once you go into sub-windows, you get some duplication, which I've tried to show with colors. And so once you have repeated haplotypes, you can store just the unique haplotypes in the sub-window and get further compression. You repeat this until, in my implementation, until you get down to a sub-window with less than 16 haplotypes. Then you stop. And it does very well. So this is just comparing to BGZip. This is on 200,000 sequenced UK biobank genomes from our phasing of the data, and just for chromosome 20. But this result will hold for the whole genome. So on the y-axis, we have the size. On the x-axis, we're looking at different size subsets. And when you double the sample size, the size of the gzipped VCF file tends to double, approximately. But the BREF4 file size only increases by about 60%, so it has a much slower growth rate, sublinear. And as a result, the chasm opens up between the different file sizes as you get larger and larger. So you get a huge amount of compression compared to gzip. And this is sort of the data slides. It hopefully will give you some intuition of how much compression you get with different methods. So we're looking at a series of methods sorted in order of amount of compression, starting with BGZip and ending with PBWT, and then following that with a gzip compression, because gzip can be applied even to any kind of data. The second column gives the reduction in VCF file size. So compared to the uncompressed VCF file, how much compression do we get? And so earlier I asked you, how much do you think we can compress phase sequence data? Whatever number you might have guessed, you can compare it to what we see here. So gzip compression, bgzip compression gives you a 300-fold decrease in file size. Amazing. But if you want to store 400,000 genomes, you're looking at 3,600 gigabytes or 3.6 terabytes. It's still a difficult amount of data to work with. So BREF3. BREF3 gives you another 9-fold decrease compared to bgzip. It gets the data to store 400,000 genomes down to about 433 gigabytes. BREF4 gives you another 5-fold. So that extra recursion that BREF4 does, I mean, it's sort of diminishing marginal returns, but 5-fold is still nothing to sneeze at. BREF4 gives you another 5-fold comparison down to 86 gigabytes. And it's important to remember this 86 gigabytes is for the whole genome. Typically, you analyze just a chromosome at a time. So we're talking about 8 or 9 gigabytes at most per chromosome, and that's all that needs to be stored in memory. So then PBWT, and you see it has very similar performance to BREF4. 81 gigabytes for the whole genome. You can get another 2-fold approximately improvement by following up PBWT with gzip, and that gets you down to 45. But BREF4 is very much in the competitive with PBWT on compression, or even PBWT with bgzip. Where BREF4 has many advantages is what you can do directly on the compressed data. You don't have to compress, or if you decompress, or if you do decompress, you don't have to decompress very much, as I'll explain. So the last 4 columns here just ask you, what can you do on this compressed data without having to decompress it? Can you count alleles? Can you filter markers? Can you filter samples? Can you query alleles? bgzip, nah, nothing. BREF3, you can do it all. BREF4, you can do it all, but with a caveat. There's a little asterisk that there's some fine print. So BREF4, because you're doing recursion and storing pointers on windows and on subwindows and on subwindows of subwindows, every level of recursion you do adds an array. That when you have to extract an allele, you have to query an array. And so that can slow things down, but there's a simple solution. BREF4 by its construction is very easy to inflate up to BREF3. You don't have to inflate it all the way and then decompress down to BREF3. You can just go directly there from BREF4, partially decompress the data to BREF3, which is a unique and very helpful property. The inflation's fast because the inflation factor is very small. BREF4 is about 14,000-fold compression compared to uncompressed data, and we're only decompressing it up 5-fold. So you're not increasing the size that much, so that makes it very fast. And then what you do is it's analogous to replacing a sequence of evaluated functions, h of x and g of h of x and f of g of h of x, with just the composition of functions of x. So what you're doing is you're taking multiple arrays and you're essentially collapsing them down into arrays of pointers into one array. And you get BREF3 format, and then you have the very fast access. And this reading in, this inflation that you perform is very fast, and it's worthwhile because the file size you're reading is 5 times smaller than BREF3, so it's faster to read it in and inflate it up to BREF3 than it would be to work directly with BREF3. So then PBWT, you can count alleles, yes. You can filter samples, at least in theory. I don't know if methods have implemented this, but yes, you can filter samples if it's PBWT compressed. But you can't filter markers, you can't query alleles, you can't store the PBWT data in memory, you have to inflate it, which means you have to spend the time to decompress and you have to have the memory in place to decompress the data. PBWT plus gzip, like I said, gives you another approximately 2-fold reduction, but like gzip at the top of this, once you gzip things, you have, no ability to do these other things. Okay, we covered a lot of ground, but I'm happy to take any questions if there are any.